Hi, Palesa. I hope you're well. I have emailed these to your mom, but I thought it might be helpful if I talk through them on video. So I'll send a couple of videos that are hopefully not too long, that sort of talks through my notes, just to kind of help you understand what's going on. And then if we do see each other again next week, then we can spend time going over papers and questions and testing your understanding and everything. I will also be sending your mom some questions on meiosis that you can look through yourself and everything. But anyway, let's just start with cell division overall. So as you know, there's two types of cell division. It's mitosis and meiosis. I want to go over mitosis first, even though the focus is meiosis, mainly because a lot of the features of mitosis are the same as meiosis, and I don't want you to be focusing, when we talk about meiosis in the next video, I don't want you to be focusing on like the mechanics of things. I only want you to focus on what's special about meiosis. Okay, so let's first start off with what is a cell cycle, and that's just talking about the process by which a cell divides. Usually there's interphase, and then that's followed by the actual cell division, which is either mitosis or meiosis. So in this case, we're going to talk about mitosis. I did a brief overview just on a little sticky note here so that we can go over it, but we'll go over it in detail. So mitosis consists of two phases. It's got the interphase, which is before the actual division process takes place, and that's primarily focused on cell growth and preparation. And then mitosis, which is primarily focused on cell division. The interphase isn't actually part of the division process. It has three main phases. And okay, let's talk about this in general. This is just a summary section so that you can look at it and just kind of get the broad idea of what's going on. Okay, so interphase is by far the longest part of the cell cycle. In fact, any cell that's not actively dividing is considered to be an interphase at any time. There are some cells that never divide or very, very rarely divide, like in the brain. So in a sense, they're kind of in interphase their whole life. But usually it's just a cell kind of preparing itself to be able to divide. And it's usually about 90% of the division process. Okay, I'm actually going to turn the camera around. I think it'll be a bit easier. Okay, so it's got three main phases. It's a G1 phase, an S phase, and a G2 phase. And it's fairly sort of, the G stands for growth. So the G1 phase, the cell is growing a lot, and it's also producing a lot of proteins. What do I do with my pen? I like to point. I'll just point with this, it's fine. So that's sort of what I'm indicating in this diagram. I'm not putting all the information in the cell. I'm just putting what's relevant to each phase. So here I've just indicated that it's growing, and I've indicated a lot of proteins inside. Proteins are necessary for cells to grow because they're the building blocks of everything. Okay. A cell is always kind of growing and adding and producing proteins. So it might suddenly be triggered to actually start the division process when the surface area of the cell is too small in relation to the cytoplasm. So as the cytoplasm gets more and more and more, there's a lot of it compared to how much surface area there is. Whereas if you've got a lot of little cells, they'll all contain in total the same amount of cytoplasm, but the surface areas in the cell membrane, there'll be a lot more of it. And that's really important for a cell to be able to function. It needs to be able to have its cell membrane. Anyway, so that's just out of interest. That might be what triggers the cell to divide. The S phase is when the DNA replication happens, just like we talked about. This is what a chromosome looks like when it's dividing. So the centromere keeps it apart. These are the chromatids, and this whole thing is the chromosome. It only really looks like this, each chromosome, once it's dividing. It's usually a single strand. So in this case, each of these will be the same genetic material having undergone division. And then the G2 phase is just a little bit more preparation. It's more cell growth, and the organelles start to replicate. They start to sort of double in number so that they can separate between two cells. Okay. All right, so I've drawn out a little bit of what happens with mitosis. As you know, I like to focus more on pictures than notes. So let's do that. Everything about the picture is also in the notes next to it. But basically, you've got your 4 main phases. Oh yeah, that's what I wanted to say as well, is you've got the process of cell division, which is called karyocytosis, where it's basically focusing on the division of the nucleus. And then you've got cytokinesis, which actually divides the cell into 2 different cells. Okay, so prophase, basically, again, I've only drawn in what's actually relevant to remember about this phase. So what happens is your chromatids and chromosomes become very thick so that they're actually visible under a microscope. The nucleolus starts to disintegrate. So does the membrane of the nucleus. And then in the meantime, these centrioles, which basically provide like fibers, there's usually just one, but it splits into 2 and starts to move across the cell. So those are the main things that happen in prophase. In metaphase, all the chromosomes line up along the equator. The 2 centrosomes are now at opposite poles, and what they basically do is that they produce these spindles that attach to the centromere between these paired chromosomes. So don't forget that each side is an identical copy of the other side of the chromosome. Okay, so that's metaphase. Anaphase is basically when the spindle fibers start to shorten and it breaks apart these chromosomes so that there's a single chromosome going to each side. And then in telophase, that's when the nucleus starts to form again, and the cell starts to divide in 2. So you can now see you're basically starting off with a single copy of each of the chromosomes. In an animal cell, what happens is it kind of pinches off in the middle to create 2 cells. In a plant cell, much of the process is exactly the same. It's just that the difference is that that they have chloroplast but the main difference is at this point of cell division is that instead of pinching off, they form something called a cell plate in the middle that eventually just develops into another cell wall. And then you undergo cytokinesis, which is when the cell actually splits into 2. And each of these is identical to the parent cell. And also, just out of interest, this is actually what DNA usually looks like. It's not in these neat chromosomes. It's usually just like a lot of squiggly mess inside the cytoplasm.